Hey guys, today we will learn about plasma proteins. Plasma proteins, you know that what is plasma? Plasma is fluid portion of blood. It forms 55% of blood volume. And proteins present in plasma are known as plasma proteins. What is the average concentration? Average concentration is 6.4 to 8.3 gram per cent, in which there are three major groups of albumin and of plasma proteins: albumin, globulin, and fibrinogen. Albumin constitutes 3 to 5 gram per deciliter of blood. Average is 4.8 gram per deciliter of blood. Globulin 2 to 3 gram per deciliter of blood. 2.3 gram per deciliter is average, and fibrinogen 0.3 gram per deciliter of blood, and range is 200 to 450 milligram per deciliter. This is the average concentration of plasma protein. Other proteins are also present, prothrombin, other clotting factors, and many things, transferrin, etc. they are present, but they are in minor amount. Main plasma proteins are albumin, globulin, and fibrinogen. Then when its level increases, when average concentration 6.4 to 8.3 gram per cent, when it increases, it may be called hyperproteinemia. This is seen in burn, dehydration, diabetes insipidus, when water from plasma is reduced. When water from plasma is reduced. And hypoproteinemia. Hypoproteinemia means when the concentration of these factors, albumin, globulin, fibrinogen, is reduced, especially albumin will reduce, and this will lead to reduction in colloidal osmotic pressure. Colloidal osmotic pressure is responsible for maintaining volume, and it will cause to, when colloidal osmotic pressure will decrease, it will cause edema. This is about hyperproteinemia and hypoproteinemia. This is average. What is AG ratio? This AG ratio means albumin globulin ratio. AG ratio is 1.7 to 1. If AG ratio is 1.7 to 1, it is very important to remember. In case of liver diseases, this albumin secretion or synthesis is reduced and globulin secretions are going on. Therefore, this AG ratio reduces, or it may be reversed. Synthesis, from where plasma proteins are synthesized. In embryo, a mesenchymal cell secretion. Mesenchymal cells secrete this protein, especially albumin, by secretion and by dissolution. Means mesenchymal cells dissolve that, and this will form the plasma protein, especially albumin. In adults, liver is the main site for formation of albumin and fibrinogen. Liver is the main site of formation of synthesis of albumin and fibrinogen. Then tissue macrophages. Tissue macrophages in liver, spleen, and bone marrow forms alpha and beta globulins, and plasma cells and lymphocytes form gamma globulins. This is the synthesis. Source of plasma protein, from where it can be synthesized. This is the site. A source is diet and cell degeneration. Diet, when we take our diet, proteinous diet, then these will form this plasma protein, a protein, and cell degeneration. That cells are being destroyed in our body, and this cell degeneration releases amino acids, and this is used in formation of the plasma proteins. Whipple's describe three types of cell proteins. First is fixed and indispensable. Fixed and indispensable means it is part of the cell. integrated part of the cell structure, and it cannot be withdrawn. Otherwise, cell will be destroyed. Then dispensable reserve protein. Dispensable reserve protein means proteins are there, and this may come in emergency in the circulation and used in metabolic process. And third is the wild reserve protein, means the proteins are present in the cell and in the plasma or reversible state, in labile state. It will go into the plasma when cell proteins are more, then it will go into the plasma protein, and when plasma protein is more, it will go into the cell. And now functions of plasma protein: coagulation of blood, maintenance of colloidal osmotic pressure, acid-base balance, viscosity, maintain blood pressure, causes immunity due to gamma globulins, and reservoir functions. These proteins are reserved in the plasma, and other metals, etc., that is combined with plasma proteins are reserved, and transport hormones, drugs, metals are transported by these metals. Transferrin is the protein that is involved in iron, and ceruloplasmin for copper transportation. Then chiral or triphones for plasma protein by leukocyte. Chiral and triphones are formed of plasma protein by leukocyte that causes nutrition of tissues. Hepatoglobulins prevents iron loss from kidney and protects kidney from hemoglobin. These are the main functions of plasma protein. Okay, in this lecture, I describe what is plasma protein, that is protein present in plasma. Average concentration is 6.4 to 8.3 gram %, and this consists of major plasma proteins: albumin, globulin, and fibrinogen. Albumin is 3 to 5 gram per deciliter of blood, 4.8 gram is average. Globulin 2 to 3 gram per deciliter of blood, 2.3 is average, and fibrinogen is 0.3 gram per deciliter of blood, and range is 200 to 450 milligram per deciliter of blood. Source of plasma protein: diet and cell degeneration. AG ratio is important. This is 1.7:1. Albumin concentration is more than globulin in normal condition. Otherwise, in hepatic diseases, it will reverse, it will decrease, or it may reverse. So, because the liver is the main site of albumin formation, and plasma in liver diseases, albumin synthesis reduces, but plasma cell and lymphocyte are making this globulin and leading to decrease in AG ratio. Source of plasma protein: Whipples describe 3 types of cell proteins. Fixed indispensable cell protein means it is the structure, integral part of the cell structure, and when cell will die, then only then it will be liberated. Dispensable reserve protein, that is in emergency it will come out, and wild reserve protein, that is in constant relation with plasma protein. So when cell protein is reduced, plasma protein will give it to the cell, and when proteins are more in the cell, less in plasma, then this will go into the plasma, making it burn. Functions are innumerate function: coagulation of blood due to coagulation, clotting factor, maintenance of colloidal osmotic pressure, acid-base balance, viscosity, maintain BP, in immunity, reservoir function for plasma protein, and metals and hormones are reserved to some extent. Then transport of hormones, drugs, metals, transferrin, ceruloplasmin. Chiral or triphones are formed from plasma protein by leukocytes. These chiral or triphones are used in nutrition of tissues. Then hepatoglobins prevent iron loss from kidney and protect the kidney. These are the main features. One thing is hyperproteinemia, when protein concentration, albumin, globulin or fibrinogen, especially albumin, reduces. it causes hyperproteinemia, COP, that is called vital osmotic pressure reduces, and this will lead to edema. May increase in case of burn, dehydration, diabetes insipidus, when fluid is lost from the circulation, and then when more fluid is lost, then it will cause concentration of this plasma protein. So this is all about, I described in detail all the main features, all the main features of plasma protein. So in next class, if you have any doubt, you may call to clear your doubt. Okay, thank you.